Yeah. So hello everyone, and it's so good to see you all again. It's the fourth of the series Big Questions Innovative Approaches, which is a series started that stemmed from Saurabh and Deepa, and IndiaBioscience is very happy to be associated with it. And we always strive to bring cutting edge research from all across the world to India, and we've had the past three talks have been very insightful and have covered diverse range of scientific topics, and have also been taken up by speakers across the world. And today we have another eminent scientist with us, Alpha Yap, whose introduction I leave to Deepa and Saurabh to take over. But I'll just leave you all with this thought that we are all very happy to have you here, sir, and we're looking forward to the talk.
Thank you very much.
Deepa and Saurabh, you can take over.
So it's an absolute delight to have Professor Alpha Yap here today with us. Alpha is a professor and group leader at the Institute for Molecular Bioscience at the University of Queensland. Many of you know in the audience, he's a global leader in the field of coherent biology. His lab studies how tissue mechanics and mechanotransduction participate in epithelial morphogenesis and homeostasis. And today, Alpha, will talk to us about his journey of collaborating across different disciplines with colleagues from physics, developmental biology, and mathematics in studying how mechanical forces are generated and sensed to coordinate cell behavior in tissues. And with that, I turn it over to you, Alpha. Absolute delight to have you here today.
Deepa, Saurabh, thank you so much for having me here. It's an honor to be here, and I confess it is totally intimidating to be speaking. I have to read this. To be speaking in a series about big questions and innovative approaches, and the reason is that, in truth, I don't think I've ever really had any big questions, and we'll see how innovative the approaches are. But what I thought I would do, if you don't mind, is tell you about a story we published recently, which I think is interesting and speaks to a long-standing problem in sort of cell developmental and perhaps cancer biology as well. And I hope that in the process, and I'm going to try and multitask and share my screens, which may go very badly, but we'll see, because I'm very bad at multitasking. So I hope that in the process, it'll give you a sense of at least how, we as jobbing experimental biologists have found our way to working, hopefully increasingly across disciplines to at least help what we do. And it's really about, how we've learned over the years to work with particularly with non-biologists, physicists and theoreticians over a range of topics. And so just a little bit of background. So my long-standing interest has been in the context of epithelial biology. So as many of you know, epithelia are some of the fundamental modes of tissue organization in the body. They are some of the earliest tissues in the embryo that therefore form a fundamental mode of tissue organization. They're a basis for morphogenesis. And of course, in post-developmental life, they form the barriers of the body, such as in this case, the gastrointestinal tract. And as such, they are the source of common diseases, particularly inflammation and cancer. And from my sort of naive perspective, one of the sort of long-standing questions really has been how the organization, dynamics, and homeostasis of a tissue sheet, a multicellular collective, is controlled and determined by interactions between the cells and their environment, specifically cell-to-cell interactions and cell-to-matrix interactions. Although we don't do anything with matrix. And as Deepa mentioned, we have for many, many years focused upon cadherin cell-cell adhesion molecules, and the minimal view of that cellular system looks a little bit like this, that what you have is, if you have one cell here and another cell here, with the actin cortices of each cell's on the internal side of the membrane, The so-called classical coherents are single-pass adhesion molecules that mediate adhesion when their extracellular domains come in contact with one another, but as well they engage with cytosolic partners through a range of associated proteins, particularly these so-called catenins that interact in a stoichiometric one-to-one fashion. And some of the key cytosolic partners include the actomyosin cytoskeleton, but also a range of other molecular apparatuses that will influence cell signaling at the membrane and potentially also membrane traffic. And I think you'll have heard much about membrane traffic from others in this series, particularly my colleague Francis Brodsky, who I think spoke to you in the last month or thereabouts. One of the, I suppose, one of the major things that happened to us, if I look back, my lab's been in existence for about, maybe 20, 25 years. The first 5, maybe 10 years of our life, we really, I guess, we're classical cell biologists, and we did what classical, sort of late 20th century cell biologists do. We look at molecules, we identify or explore candidate molecules, look for interacting partners, endeavor to try and have develop a sense of how these machines might work. But I confess that probably about 15 years ago, I kind of started to get a little bit frustrated or dissatisfied with that approach. And really it was because of the challenge of dealing with the sheer complexity of cellular biology, right, of cellular apparatuses, where what is dealing with are multiple molecules, particularly in vertebrates, multiple isoforms, and the difficulty is trying to identify ways of parsing that complexity, especially when you have lots of feedback and redundancy. And I was actually inspired by giving an undergraduate lecture one day about adhesion. And a student afterwards came up and said, what's adhesion? And, my first response was, dear me, how naive you are. But actually, when I started to think about that, it was an important question to revisit. Because, of course, what adhesion does is that it holds particles, objects, things together by resisting forces that might pull them apart, that might disrupt that interaction. But an important question really is, what is the source of that force? And in the when I was a student, a postdoc, and so forth, often the way we would think about the forces that cell adhesions resisted were these large-scale forces of friction and encounters with the environment, forces that you might describe as being extrinsic to at least the cells involved and perhaps as well the tissues that are involved. But what we've come to realize through partly our work, partly the work of many others, such as Tamala Kui and C.P. Heisenberg and so forth, is that as well as these large-scale external extrinsic forces, there are a variety of intrinsic forces that are essentially generated by the cells exerting forces, particularly tensile or contractile forces, exerting those forces against one another. And you can reveal the presence of those forces in a number of ways. One of the easiest ways is simply to take a laser and cut a junction. What you see is when you cut that junction, there's a recoil implying that there was a tension that was pre-existing in the junction before you cut it. And if you make larger scale cuts in a monolayer, cultured monolayer, what you see is that there's a pattern of recoil that extends or translates or amplifies to the supracellular scale, suggesting that that even under apparently quasi-static conditions, when nothing very much is happening that's too dramatic, these cells are constantly exerting tensile forces upon one another. And of course, what you will know better than I is that that tension reflects the physical and dynamical coupling of the adhesion system to the actin and myosin cytoskeleton. which, through its capacity for actin binding and myosin filament sliding, can exert or generate contractile forces within cells. And so over the years, we've been engaged with trying to understand how those contractile forces are regulated and how forces might be sensed. But I guess more broadly, what that's come to make us think about is the idea that the generation of physical forces by cells at their contacts is kind of like a handshake. And in the same way that we exert forces when we shake hands with another person, we also infer things about that person. Shaking hands is a mode of communication that we interpret rightly or wrongly. A light handshake, a hard handshake, a wet handshake, blah, 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 blah. And so we've started to wonder whether there are mechanisms by which cells not only exert force upon one another at adhesions, but might also sense those forces to exert some kind of biological outcome, to serve some kind of biological function. Now, this function and we've sort of explored a number of functions over the last 5 or 8 years. The function I'd like to tell you about today is really a very old biological phenomenon, which is the phenomenon of contact inhibition of cell migration. And this goes back to the 50s with, the pioneering work of Michael Abercrombie and Joan Heisman in a variety of cells, mesenchymal cells, as well as epithelial cells. And in our system, which is a very simple-minded system of epithelial cells, that MCF7 memory epithelial cells that are grown to confluence behind the barrier. And when we remove that physical barrier, they then migrate towards one another to close that barrier, sort of like a pseudo wound healing assay. And one sign that they have inhibited their motion, their locomotion, when they migrate towards one another and come into contact, is that if you take two populations of cells, in this case identified with LIFACT tagged with different colors, then what you see is that even after they have formed what seems to be now a confluent monolayer, if you look at the colors, you can see that there is an interface, a relatively straight interface that actually marks where they first came into contact with one another. So that's one of the signs that we think that there truly is contact inhibition of epithelial migration occurring in our system. And this phenomenon of contact inhibition of migration has been implicated in a variety of homeostatic processes, including wound healing, as well as tissue turnover. And ever since the days of Abercrombie, it's been thought to be potentially perturbed in aspects of carcinoma and cancer. But having said that, one of the fundamental questions in this problem is really the question of how do migrating cells know when to stop? If it's contact that's involved, then presumably it is something that is presented on the surfaces of cells. And of course, if like me, you're interested in cell-cell adhesion, then you've kind of your prejudice is to wonder whether or not cadherins might be as the first receptors or some of the early receptors that mediate contact, whether they might be involved. And indeed, again, over the decades, there have been a variety of studies that have endeavored to try and identify a role for cadherin, and more importantly, to try and understand what might be the cellular mechanism that could be engaged. And just to remind you again of the minimal complex, because we'll come back to this in a little bit, a number of proteins that interact stoichiometrically with the cytoplasmic tail of the cadherin. One of them is the famous beta-catenin. We treat it for our purposes as a linker to alpha-catenin, which ultimately can bind to actin filaments. And that's going to be important as we get into this. So as you might imagine, we started to get interested in the idea that perhaps adhesion and perhaps forces generated when cells make adhesion might be relevant for understanding cardiac inhibition. And the way we normally start a problem is really just to do some experiments and hope something interesting will turn out, and that we recognize that something interesting is turning out. And so we meaning Eva and Ortstra, started to look at these wound healing experiments. And, you know, as you imagine, cells migrate to one another, and within a relatively short time of coming into contact with one another, they start to make adhesions. And the adhesions initially looked like these punctate structures that progressively get wider to eventually, after several hours, make something that looks like a relatively mature adherence junction of zonular adherentes. And I guess importantly from our point of view, it soon became apparent that indeed the literature and our colleagues were right, that this phenomenon of cardiac inhibition did require the function of the cadherin complex. Because if we take wild type cells, then what we see is they make these nice, relatively straight interfaces with one another. But if we knock out alpha-catenin, which, as I say, is a key component of the adhesion complex and couples to actin, then what we see is that instead of forming these nice, relatively linear interfaces, they seem to interpenetrate as if they were essentially unable to see one another and just kind of migrate inwards to form these projections into one another. And if you measure how far they migrate in, what you see is that indeed that interpenetration is increased, suggesting that that their ability to recognize contact as a signal to stop moving might be compromised. So as we start to get into this, made us think, yes, indeed, there was some elements that could link, a molecular complex that mediates adhesion to the inhibition of cell locomotion. But in a way, that's just rediscovering what many others in the field have identified before in different fashions. But there's kind of a subtlety here that I think is important, and that really I have always found rather puzzling. What we're dealing with are moving sheets of epithelial cells, and those sheets are connected, or the cells within those sheets are all already connected to one another as they move. As it were, if you imagine that there are two populations of cells, these are going to connect head on as they close the gap. But those cells, including the leader cells that are guiding, or at least, as it were, leading migration, those cells are connected to one another by adherence junctions. And just for simplicity, we refer to these junctions as side-to-side contacts, as opposed to the head-on contacts that occur when the populations meet. And so, this simple observation just raises the question, why is it that locomotion is inhibited only when cells make head-on contacts with one another? Or put another way, why is it if they're making side-to-side contacts, they aren't inhibiting locomotion? It can't be simply, or it's unlikely to be simply a matter of their making adhesions and ligating, because there's no reason to suppose that these adhesions, at least as sticky events, are necessarily much different from the head-on adhesions. There must be something else, we thought, that is distinctive about a head-on contact, that and different from a side-to-side contact. There must be some property that was being sensed by the cadherin. And that's really the story I'd like to tell you about. And the we, when I say we, is actually has nothing to do with me. It was, above all else, Eva Nostra, who's a fantastic postdoc in the lab, who drove almost all the experimental work with some help from others in the group, and our collaborators in theory, Pere Roca-Cusacks and Mario Hermoso, who are in Barcelona, with the help of a number of our colleagues, both in Australia and elsewhere in the world. And the story is now, recently published, and so the gory details are there. So I hope I'll spare you most of those gory details and just tell you a bit about the experimental logic. So, how did we, i.e. Eva, try to dig into this problem. And, the first thing that we did was, or what Eva did, was really to look more closely at the movies that he was making. And here you have two sets of cells that are migrating across the gap to make contacts with one another. In this case, they're both expressing life act with different colors. So you're looking at the actin cytoskeletons. And there are a number of things that I hope you will perceive as this movie loops over. The first is that they move forward by making protrusive lamellipodia, sheet-like extensions. That's nothing very remarkable. Those lamellipodia are the regions of the migrating cells that first touch. They overlap, and I'll come back to this in a moment, and eventually the dynamic protrusive activity of those lamellipodia go down. The other thing I hope you'll see here is that they have prominent retrograde flows of actin. This becomes really important. Forgive me for belaboring the point. If you just look, actually, I'm just going to pull this back here if I can. If you just look at these lamellipodia and look at the actin, the lamellipodia is going forward, the actin is going backwards relative to the front of the cell. And so that's what I'm going to describe as a retrograde flow of actin. And just to belabor the point, I'm going to ask you to remember that this cell moving upwards, the green one, has a retrograde flow that goes towards the bottom of the screen. The guy on the top has a retrograde flow that goes to the top of the screen, so that the retrograde flows, as the cells come into contact with one another, are anti-parallel. I'll show you that again in another moment. As well, the flows stop upon contact as the cells make contact with one another. So just to reiterate, fundamental features that Eva was seeing is that the lamellipodia are the basis for adhesion. Lamellipodia overlap initially, and as they overlap, adhesions or coherent adhesions form, and the adhesions are important for inhibiting the dynamic activity of those lamellipodia. And one way in which you can judge that dynamism is to make sort of a chymograph across an interface. If you look at the chymograph with time in the horizontal axis, you'll see that both of these cells, they protrude, retract, and eventually will stabilize. But if we take cells that can't make productive adhesions, i.e. alpha-ketene knockout cells, instead of the dynamics reducing, they basically continue to be dynamic throughout the length of our movies. So what that suggested to us was that the inhibition of lamellipodia might be important. First point of contact, these are the organelles that are important for guiding and driving locomotion. So somehow the cadherin, upon adhesion, was able to inhibit lamellipodia formation. The question then was how? As we started to think, or as Eva started to think about how, he started to wonder about the importance of retrograde flow. And that's because, Eva reads the literature in contrast to people like me, and he realized that the retrograde flow in lamellipodia is a key factor in the locomotor apparatus, because it can generate forces for protrusion, particularly when the retrogradely moving flow of actin engages with the adhesive interface with the substrate. So this is the model that Lindsay Case, Claire Waterman, and Garden Sanusa developed some years ago. The idea that frictional interaction between the integrin complex and retrograde flow would retard retrograde flow, and therefore cause the flow to effectively generate a forward protrusive force. So we wondered, was flow going to be something that could be interesting to us? And perhaps the next surprising observation was the finding, or Eva's finding, that even when cells overlap with the lamellipodia, the flows in those lamellipodia don't stop immediately. They keep going for a while, 10, 20, 25 minutes. And as I mentioned to you before, the flows in those overlapping lamellipodia are oriented away from one another. So they're anti-parallel. And what was notable is that the assembly of adhesions, cadherin adhesions, seemed to occur a little before the flows stopped or the lamellipodial dynamics stopped. So we wondered, again, whether perhaps there was an impact upon flow that could be relevant. So we were sort of starting to hypothesize that perhaps the cadherin was inhibiting the cortical flow that's present in the lamellipodia. And by doing so, if the cadherin could inhibit cortical flow, then that could be a way in which you could inhibit lamellipodial activity and protrusion. And as we started to think about how an adhesion could inhibit flow, we were struck by this anti-parallel orientation of the contacts, what I've belabored before. And we started to wonder, could it be that we have a mechanism that is something like this? Imagine that if each of those flows is like a team in a tug of war, right? Then when the cadherins make adhesion with one another, when they productively bind to one another, effectively what they could do is that each would couple to its actin cytoskeleton, and they would actually mechanically couple the flows with one another. And because the flows are directed in opposite directions against one another, they would effectively be like a tug of war. They'd be pulling on one another when they were mechanically coupled, and potentially that pulling could cause them to reciprocally inhibit one another. Each would pull on the other, and so effectively they would reciprocally inhibit themselves, and thereby inhibit lamellipodial formation. So that was the hypothesis. Now, if an idea like this has any legs to it, it makes a number of predictions. The first is that the cadherins must couple to retrograde flow, right? Otherwise, I'm wasting your time and Eva's. And indeed, they do. And the important clue that came to tell us that this might be the case really came from looking at the lamellipodia before contacts were made. And what Eva saw was that even in the lamellipodia, he saw populations of cadherin. And I'll cut to the chase. Some of those populations are in vesicles, and they are the green ones here. Some of them are actually on the surface, and they're the white ones. So, even before cells make contact with one another, at least 50% of the cadherin in the lamellipodia is on the surface. And if I could show you the movie that decided not to work in Keynote today, what you would see is that these two populations of cadherins show different movement. The ones that are vesicular show a rapid backwards and forward movement that is consistent with microtubule-based transport. But the ones that are on the surface show this retrograde movement from the front to the back that's slow and processive, and looks rather like the retrograde flow of actin that I showed you before. So that starts to make us think that even before cells make contact with one another, there is E-cadherin on the free surface of the lamellipodia that's coupled to the cortical flow, so that it would be poised to mechanically couple the flows of the contact cells when they made adhesions with one another. Now, the other prediction is that when these lamellipodia overlap, when they make adhesions, the flows must be mechanically coupled to one another, right? Otherwise, this mechanical reciprocal inhibition wouldn't work. And the way that Eva decided to try to test that idea of coupling, well, he tried a number of things and you'll see them in the paper, but perhaps the most elegant was this experiment. What he did was say, well, if the flows are coupled, they probably require alpha-catenin, and indeed they do, or these alpha-catenin is needed to couple the cadherin to the flow. Now, if they're coupled across a contact, then I should be able to uncouple the process if I knock down alpha-catenin. Sure, that works. But he said, more importantly, if I knock down alpha-catenin in only one cell of a pair or one population of a pair, that would be sufficient to uncouple, because you would need connection to actin on both sides of the newly forming junction. So that's what he did. And the experiment basically is to take cells that have either wild type that have alpha-catenin in them, which is the cells in blue, or cells in which he's knocked down alpha-catenin. And what I'm going to ask you to do is just to really look at this movie. Now, this movie shows E-cadherin only in the cell that doesn't have alpha-catenin. And normally, what we would see is that in the lamellipodia, these clusters of cadherin would move into the body of the cell, would move retrogradely. But what we see when we do this uncoupling experiment is that now these clusters of cadherin are moving in the opposite direction. And we think what's happening is that they are being pulled in an antegrade or opposite direction by the flow in the wild type cell. So that suggests to us that indeed this system is uncoupled. The key question is what happens to the dynamics of lamellipodia in this asymmetric uncoupling. And the answer is that the dynamics of lamellipodia are ignorant or become blind to contact. The lamellipodia keep on protruding and retracting, and they never stop as they do in the wild type situation. So that suggests that indeed that potentially this model of a mechanical tug of war would work. It started to make us think about other things. It made us think that, well, you know, if there was a mechanical tug of war, what that would do is that it would apply or generate, or yes, it would generate or apply a tension to the cadherin complexes that are connecting these two flows that are moving away from one another. And that, of course, started to make us particularly interested because the cadherin has a variety of mechanotension sensitive apparatuses in it. And one of the best understood is alpha-catenin, because alpha-catenin can undergo conformational changes to unfold when tension is applied. There are two that are well known, one of which involves the central region of alpha-catenin, and a second of which, less well known, involves the actin binding domain. And so the first thing that Eva started to do when this became a possibility is to say, well, does indeed, can I see tension being generated preferentially at head-on contacts? And he used a variety of ways to get at that, but including antibodies that detect the unfolded tension-sensitive conformation of alpha-catenin. One of them is the one that is seen in the actin binding domain. And indeed, if he looks at the intensity of the signal for this tension-activated conformation, it's higher at head-on contacts than side-side contacts. So that started to make us think that perhaps there could be a mechano-sensing and a mechano-transduction event that was involved, as well as simple sort of mechanical coupling of flows. And that became very exciting for us because the application of tension to alpha-catenin, particularly tension in the C-terminal actin binding domain, is thought to engage a catch bond between alpha-catenin and actin. So as you guys know, I mean, a catch bond is simply a bond that shows has the interesting property that its lifetime increases when you apply tension. So engagement of a catch bond between alpha-ketene and actin would mean that the interaction between actin and alpha-ketene, and hence the adherent complex, that interaction would get longer and effectively stronger as you apply tension. And we thought this was a beautiful idea. Well, actually, I thought this was a beautiful idea because, what that would mean is that when we coupled the flows and tension was being exerted, then the strength of binding of the interaction between the adhesion system and actin would be increased, and that would actually enhance mechanical coupling, and therefore it would be a beautiful way to proportionately increase mechanical coupling to inhibit lamellopodia. And this, I thought, this seemed to me to, this is my fault. This seemed to me to be a beautiful idea. But when Eva started to think about this a little bit more, he said, look, conceptually, qualitatively, plausible. But it's ultimately a quantitative question. And we really need a way of analyzing this quantitatively and framing the problem quantitatively to test whether, in fact, the idea is reasonable or plausible or possible. And this is where we turn to Perret, because Perret has really been the pioneer in thinking about how engagement between adhesion molecules and actin can be conceptualized as a clutch. And so what the way I think about a clutch is that it is a reversible interaction between, in this case, an adhesion molecule and actin. And by virtue of being reversible, it will have an on rate and an off rate. And in the case of a catch bond, then the off rate will be decreased by the application of tension. And so we reached out to Perret and Mario and said, look, you guys have been modeling these clutches in the context of a system where, there's adhesion and actin. Could you model them in a situation where that adhesion links to actin on the other side of the contact? And would the idea work in this system? Would a catch bond be able to generate sufficient mechanical coupling so that the flows would be inhibited using sort of what we know about on and off rates for the molecules in this system, as well as what we know about forces generated by flow and so forth. And so Perret and Mario said, yep, I think we can do that. They went away and did that. And they came back and said, you know what? It doesn't work. What their simulations showed was that on very, very short timescales, timescales of fives of tens of seconds, yeah, it can work insofar as the engagement of a catch bond engagement of adhesion and engagement of the catch bond will slow flows. But on the timescales that we saw experimentally, which are timescales of tens of minutes, that isn't sustained. And the intuition that they tried to explain to me was this, that even when you engage the catch bond, even when you enhance the lifetime of the bond between alpha-ketene and actin, the off rate is still occurring on timescales of seconds. So they still disengage on rapid timescales, so they can't build up sufficient mechanical coupling to actually slow and reciprocally inhibit their flows in any sustained fashion. And you can imagine that when they said that to us, this came as kind of a bummer. But then they smiled and said, having said that, we did play with the model. And there is a circumstance we've played with a number of things. We've changed off rates and so forth and so forth, and it doesn't really work. But there's one circumstance which it can work. And it could work if, in addition to increasing the lifetime of interaction with actin, if the application of tension were also to recruit cadherins into clusters. So, what I mentioned to before is that we see these clusters on the surface that we think are the initial points of contact. What Pere and Mario said is that if the clusters grow in size, if you add more clutches to the system, then under these circumstances, you have increased rebinding of adhesions, and indeed you can start to generate sustained forces upon one another that are potentially sufficient to inhibit or to reciprocally inhibit flow, at least in their simulations. So this I think was in a way the beauty of working with people that think quantitatively, analytically, and don't share your preconceptions, because they can make predictions, or their work and models can make predictions that you didn't think of. So that kind of made us think, well, is there a mechanism by which applying tension to the actin binding domain could not only engage a catch with actin, it could also promote growth of the cadherins? And thankfully there was work that had been done by Noboru Ishiyama a few years ago, which showed that the unfolding of the actin binding domain also unfolded a helix that could promote dimerization of actin, of alpha-catenin, potentially bundle actin, and potentially, therefore, cause the cadherins to incorporate into clusters and for those clusters to grow. And so if that's true, so Eva decided, well, look, I need to test this. And he said, I need to test it in a way that's as simple as possible. I don't want the complexity of dealing with contacts between cells and one another when cadherins engage in adhesion, because then you activate signals and a lot of stuff could be happening. So he said, the way I can do it is, I know that cadherins form clusters already, even before adhesion on the free surfaces of cells. So I can use that as an assay, eliminates the problem of transligation, and I can measure the cluster size by, for example, the fluorescence intensity in my system. Now, he said, what I can do is I can then knock out alpha-catenin, replace it with a mutant in the C-terminus that behaves like an unfolded tension-activated molecule. And what he found was that under those circumstances, even without applying tension, that the size of the cadherin clusters increased. But that increase was lost if he deleted that region of the actin binding domain that mediates dimerization, the so-called beta helix motif. And importantly, not only did this region support cadherin clustering, as Pere and Mario would have predicted, but it inhibited lamellopodial activity. So if I ask you just to look to the right of the screen, this is this lamellopodial inhibition assay that we've shown before. Basically, dynamics decrease in wild-type cells as they make contacts with one another. But that inhibition of the lamellopodial activity doesn't occur if we delete the dimerization motif, the beta helix motif in the actin binding domain of alpha-catenin. Now, what you'll say is, ultimately, well, that's fine, but we started some lifetime ago, we started by thinking about cardiac inhibition of locomotion. Is this relevant? So Viva went back to his migration assays, and he looked at measuring the formation of or the interpenetration of interfaces between populations. This is the control that I showed you before. And what's exciting is that what we find is that if we make limited mutations in alpha-catenin that block the capacity to promote clustering, that the contact inhibition is compromised and the cells sort of migrate past one another and the populations interpenetrate. So that started to sort of give us a model where we were starting to think that the basis for contact inhibition, the first signal, might be a mechanical signal that occurs when the cadherin couples flows that are directed in opposite directions from one another. This generates a tension in the cadherin to activate the actin binding domain or the C terminus, and ultimately this will reciprocally inhibit the flows to initiate the cessation of locomotion. But again, let's come back to this question that we posed originally, that, why is it this is specific, or apparently selective for head-on contacts? Why doesn't it occur at the side-side contacts, which is where the cadherin seems to be ligating? And again, the clue comes from the observation that I showed you before, that when we estimate tension in the cadherin or in alpha-ketinin, it's lower in the side-to-side contacts than in the head-to-head contacts. And why might this be so? And what we'd like to suggest is that it has to do with the orientation of flows. Because if this is a side-to-side contact, these two cells are leaders moving downwards. In both those cells, their retrograde flows go upwards, but they go in the same direction. And indeed, if you look at chymographs, if Eva looks at chymographs, indeed flows are in the same direction. So in a way, it's like if we were linking arms and walking along with each other, the tension in our arms would be less than if we decided to link arms and walk away from and pull away from one another. So in a sense, what we'd like to think about is that, the reading, the information that's being sensed is really not to do so much with the cadherin. The cadherin is just a way in which the cells can ultimately interpret the topology of the guys with which they engage. It's a way in which they can decide whether the cells that they are engaged with are moving in the same direction, or they've collided head on, because the topology of flows and the tension that's being generated in the cadherin will be different. And that's in a way what we would suggest might be the ultimate parameter of the cell that's being sensed, or that the cadherin uses to decide whether to stop moving or not. Okay, let me close with last 3 slides. When I think about this, what have I learned? And this is, in a way, this was an easy collaboration because Pere and Mario are biologists who do theory as opposed to physicists. So it was easier for us, if not for them. But as we've worked with theoreticians, of a number of projects in the last 5 to 10 years, I think that I at least have learned many. It has improved me in many ways. One of which is that it forces us to really define the key elements in our models. We have to be specific. Even if, as cell biologists, we know it's complicated, and our instinct is to say it must be more complicated than these limited elements. Working with theory forces us to commit to limited elements, to ask, could these be enough? It forces us to be quantitative, which I can't do. And in a way, it's most powerful when, in fact, it says that our intuition is wrong. There's something else we're missing. And that's something that, has occurred often for me. These are collaborations. And I think that what we've experienced, I think, is what everyone experiences, that it takes a lot of time and effort to make the experience work. You spend a lot of time discovering that you use the same words to mean different things. It's a lot of time trying to work out how to speak together. And I think the important thing actually, ultimately, is to work out what's the underlying intuition, right? the theoretician, the physicist, has to try and work out what is the biologist's intuition and vice versa. And hopefully in doing so, we find a way to frame the problem that can be tackled from both ends. Okay, last slide of all, you've had enough of this. These are the people who do all the work. I am incredibly grateful to them because nothing would be possible without them. And I have learned so much from the example of their talent, dedication, and assiduity. And thank you for your patience. And I'm gonna stop sharing, and I'm gonna try and move the screen back and hopefully take any questions that you have for me.
Thank you, Alpha. That was fantastic, actually. It was quite brainstorming, and especially the last couple of slides also, where you suggested like how to collaborate and what should we learn from this kind of collaboration. There are a couple of questions. Already questions are coming in. So the first one is, during cell migration event, how does few cells identify themselves as a leader cells, or is it something like the cells which are at the periphery or the boundary, they continuously look for something to touch and keep migrating towards the end? So
I think that's an incredibly interesting question. It should be said that that there has been a debate, and I think probably more in the vascular biology field, about whether the leader cells have a distinct fate. And I think, though, you will know better than me, I think the idea is coming around that perhaps there is no fate, that leaders and followers can potentially change position. We haven't seen that in our system, but that may be because we're dealing with epithelia that are relatively slow. As you say, the question is, when you make a wound, when you release a physical impediment, what is it that polarizes leader cells? And because they do polarize, ultimately, they will have lamellipodia in one direction. I think I don't actually know the answer to that. My suspicion is that it's probably a combination of two things. One is that you have a free edge, and all free edges have the capacity to assemble up to 3. More importantly, why don't you make free edges elsewhere? Why don't you make free edges at the back? And there's certainly evidence to suggest that there are small lamellipodia elsewhere, but they're smaller than in the front. And my guess is it has something to do with the fact that they do make adhesions with one another, albeit less tense ones than when they collide head on. But it's a great question.
Yeah, the second question is mostly pertinent to your first part, where have you also knocked out any other, like beta-catenin, for example, and seen any effect with the similar effect as alpha-catenin?
No, we haven't. We've tended not to touch beta-catenin too much. Firstly, because obviously you have secondary effects with Wnt signaling, but also because it seems to be a high affinity interaction that is stoichiometric, and we figure that if we were dealing with an interaction with actin that was regulated, that it's the more proximate event would be at the level of alpha-catenin, which is why we focus on alpha-catenin. Dirty little secret that I shouldn't mention in public, but I will, is that we're terrible. We often just treat beta-catenin as a linker. I know that's terrible. That's bad of us. And we will mend our ways one day.
Satyanarayan is keen to know, like, how do you define E-cadherin pools, like cytosolic versus surface pools? Like, is there any levels which affect the contacts inhibition or something like?
Okay. So the way that technically the way we identified them, something I overlooked, but basically the way Eva did is he took cells first of all, he engineered cells by CRISPR so that the cadherin had a C-terminal GFP tag. So the whole population, the whole cellular pool is GFP tagged. And then in live cells, he added an antibody directed against the extracellular domain. So he surface labeled and did secondary stuff and so forth. And so what that meant was that where he saw two colors overlap, both the surface label as well as the internal GFP, he interpreted that to be cadherin on the cell surface, whereas the cells that, the clusters that were just GFP and weren't surface labeled, he interpreted to be vesicular. Now, in our hands, it's about 50-50. We haven't looked for regulation of those pools. What we do find, for what it's worth, if you look at the pool size in a quasi-static mature monolayer, it's probably more like about 90% surface and 20% and 10% internal cytosolic and vesicular. It presumably reflects just stabilization upon formation of adhesion. That sort of goes back in sort of the dark days, the prehistory of the cadherin field when I was a postdoc back in the last century.
I'm just for the sake of time, I'm just clubbing a couple of questions here. Like, is there like you see that when the anti-parallel pool is happening, that there is a disintegration of adhesion junction itself, or the second continuation of that is like how this directionality switch rate, like do you know anything about the switching rate? Because when the cells starts switching their sides of migration, how these dynamics work. So
okay, so first question, we haven't looked to see whether, because the way we do the experiments, we grow the cells against a barrier, so and that the barrier is inert, so in a sense the leader cells are already in a way preformed, right? Because they will be basically not making adhesions with anyone else. We haven't looked to see whether the dynamics changes in the follower cells, and that's an interesting question. In terms of the orientation of flows, I guess one way I would interpret your question, which may not be correct, is that we're looking at flows that are head-on, right, which are at 180 degrees to one another. Our prediction is that there must be some sort of critical angle where this inhibition does or doesn't work, but we haven't been able to make an experiment work to test that critical angle.
So can I sort of ask a continuation to that? So if you were to disrupt actin flow on one side, how does that affect the formation of these inhibitory contacts? So basically my question is, do you really need this anti-parallel flow on both sides, or is it just incidental to the cells coming together?
So technically extremely difficult to inhibit flow fully by sort of molecular genetic approaches. So we haven't done that. I guess I would say that coming back to the asymmetric experiment that Eva did, right, where even though the flows continue, what he did was prevent one side from coupling to flow. What he found was that the formation of the growth of adhesions was perturbed, was blocked. So, our feeling would be that the coupling to flow is probably essential to generate a tensile signal that's enough to then activate alpha-ketene and so forth. And we would argue actually that, and that would promote growth of the cadherin. And we would argue that at least initially, the antiparallel organization of flows is what generates a tug of war force to start to slow the flow. Almost certainly there's more that's happening that would include changes in actin assembly and so forth, but we haven't gone there yet.
Okay, maybe the last question, question we can take. It is something parallel system than what you described, but in the brain, basically, when the neurons form different connections, like how does it work? Like, do you see something similar happening over there in the context of like forming new circuitry systems between different neurons?
I don't know the answer to that. That's an incredibly interesting question, because many years ago, Takeichi, I think, was looking at at the formation of axonal spikes, right? And that was alpha-ketene dependent. Honestly, I do not know the answer at all. My guess is that, well, I'd be open to the idea, and it doesn't need to be flow. All this model needs is to have cytoskeletal forces that are going in the opposite direction to one another. So it could be pulling through contractility, as would occur with bundles and filopodia.
Great. Great to have you, Alpha, again. And so it's really wonderful to talk to you and to listen to you with exciting science over here. And to all the participants over here, like, if you have still more questions, you can write to us and we will try to forward it to Professor Alpha and get back to you with the answers. And we won't have a session in the November, but in December, we will come back with the new session and you will soon find the announcement of that.
And feel free to write to me directly. So happy. Thank you so much. I'm honored to be part of this.
Thank you so much, Alpha. That was an absolutely fantastic lecture.
Thank you very much. Take care, everybody.
Bye-bye.